All right, let's get started. So this is our fourth lecture, and today we're going to revisit Gregor Mendel's experiments, and we're going to look at his set of experiments that were, really revelatory in our understanding of the discrete nature of inheritance. And we'll talk about the two fundamental insights that Mendel had. So that is the law of equal segregation and the principle of independent assortment. And we're going to use the rules of probability to understand Mendel's findings and the underlying model. Okay, and then finally, we'll talk about how a bit of the history of Mendel's discovery and its rediscovery, and then its implications for understanding human disease. So who knows something about Mendel? Well, I'm going to launch a little poll here and see what happens. So the question is, what was Gregor Mendel's profession? Does anyone know? Many of us know him as a monk. Many of us also know him as a botanist. Less of us know the fact that, in fact, his aspiration in life was to be a high school teacher, but he wasn't able to pass the high school teaching exam. In fact, he failed it twice. You must have been pretty good to be a high school teacher in Vienna in those days, which is where he was trying to get that job. He was actually, a really interesting guy, an intellectual. He had actually studied physics. He'd studied with Doppler of the Doppler effect in Vienna. He had aspirations to become an educator, and ultimately he became an Augustine monk in the town of Brno, which is in what is now the Czech Republic. And he was of course, a researcher. And if you want to learn more about Gregor Mendel, I really recommend reading this book called The Monk in the Garden, where it talks about his life. I read that a couple of years ago and really, really highly recommend it. Gregor Mendel, of course, he was a monk living in a monastery. The monastery had a beautiful garden out the back, and he was a botanist. And he wasn't just a tinkerer. He wasn't just, growing things for the purpose of eating them or for the purpose of growing beautiful flowers. He was truly a researcher, and his organism of study was the pea plant. It's actually relayed in this book that the motivation for these studies was economics and agriculture. So agriculture, of course, was the primary mode of economic activity in the 1800s in Europe, and being able to breed better agricultural crops, there was a lot of financial and economic incentive to do that. And so that was, what he was doing was not uncommon and had this underlying motivation. And so he was studying the pea plant, and he looked at 7 specific traits of this plant. And so he looked at traits such as whether the seeds from the pea plant were round or wrinkled, whether they were yellow or green, whether the petals were purple or white, whether the pods were inflated or pinched, whether the pods were green or yellow when they were unripe, whether the flowers were at the termini or were axial, and whether the stems were long or short. And so these are the 7 classic traits when we think of Mendel's experiments that he used to study the inheritance and transmission. And there's one key property of all these 7 traits that was really key to his observations. And what is that property? How do these traits differ perhaps from the other trait that we're talking about, the only other trait we've been talking about in humans so far, which is height? What is the fundamental difference between human height as a trait and the traits that we're looking at here? Binary, okay? That is the key point here. They're either or situation. So height is not an either or situation. You're not either 170 centimeters or 172 centimeters. There is a continuous distribution of heights. And that is in contrast to these traits where you either have a wrinkled seed or a round seed. You either have a yellow seed or a green seed, okay? And so that's really key here is the discrete and binary nature of these traits. And so why is the P plant the method of, or an ideal organism in order to undertake these studies? And the reason for that is the following, is that there's an experimental method that allows one to be able to manipulate or perform genetic crosses. And so here is the structure of a pea flower. And what we see here are the ovules indicated in green down here. These are the female gametes, and the pollen, which is up here. Here's the pollen, here's the ovules, and here we have, oh, sorry, this is the stigma which receives the pollen. This is the pollen is produced over here. And the pollen is the male gametes in a plant. The ovules are the female gametes. And so within this single plant, we have both female and male gametes that are produced. This makes it extremely useful because that allows us then to be able to experimentally perform genetic crosses in which we can take the male gametes and we can introduce them either to the female gametes of the same plant just by taking these pollen here and brushing it onto the stigma which receives the pollen, and then the zygote is formed here by fusion with the ovule. Or we can take the pollen here and we can go to another plant and we can do the same thing. And so we can do both a self-pollination or a self-cross, or we can do an outcross where we go to another plant, another individual, and we pollinate that plant. So this is actually practically done and continues to be done today using the advanced technology of a paintbrush. So you just take a small paintbrush, and here this individual is using this paintbrush to be able to acquire the pollen at the end of the paintbrush. There it is. And now you just can either apply that pollen to, which are the male gametes in this case, and you can apply it either to the same plant or to another plant. And so this is an amazing property of this model system, the ability to both do a self-cross or an outcross, and that turns out was very essential in this case. Can you do this particular experiment in humans? You cannot. What can you not do?
We can't self-pollinate because like you either have like, well, there are like people with like some both male and female like organs and stuff like that, but in generally like you as a female, you're not going to have the male organs to self-pollinate, I don't know, to self-pollinate yourself in a way.
Right, you cannot, we're pretty limited as a species. We can either produce male gametes or female gametes, but it has not yet been recorded in human history an individual that can produce both female gametes and male gametes, both a sperm and an egg. And thus it's not possible to do this in humans. Are there other organisms where it is possible to both self the organism and to perform an outcross? Does anyone know of an organism? Can you do this with Drosophila? I see one suggestion. Suggests that C. elegans. What is C. elegans? Right, a nematode. Cenorhabditis elegans is a model system that's actually used at NYU by a number of researchers. There are a number of research groups at NYU that perform genetic studies using the worm C. elegans. And that was identified as a model system by Sidney Brenner in the 1960s and 70s. And it had the same property as Mendel's model system, the ability to be able to do a self and to outcross. And this makes it an extremely useful model organism. All right, so, the organism I work with, budding yeast, you can also do this. But by contrast, Drosophila or mice, other model organisms that are typically used, cannot do this. So this is one of the great advantages actually of plants in general as a model system for being able to do experimental genetics, is the ability to do this selfing. Okay, so now I want to talk about the first experiment that Mendel did. So let me describe that experiment for you. So here's the first experiment that I want to talk about, and Mendel's first experiment. So we'll just use one of the examples. which is the case of yellow seeds and green seeds. And so here's the experiment, which we'll call a monohybrid cross. So what did Mendel do? So first, Mendel started off with a parental generation, and we'll denote that with a P. And he had plants that had yellow seeds, and he had plants that had green seeds. And these plants had an important property, which is that they were both pure lines, which means that they bred true. And what does this mean? This means that if you self them, you only get yellow seeds when you self a yellow plant. And if you self a green seed plant, you only get plants that have green seeds, okay? So that means they breed, we use the term that they breed true in that case. These are actually bred true, which is the past tense, so I'll use breed true, okay? And this is an important, really important property of these plants. Okay, so now what did Mendel do? He took these yellow seeds and he took the pollen, which is the male gamete, and we'll use the following symbol for males. And he introduced them to a plant so that of the green seeded plant, and so these were the ovules, and these we'll denote with this symbol. These are Greek symbols for male and female. Does anyone know what the male symbol, where that derives from?
Is it Mars?
I think it's Mars, right? This is Mars's spear. And then the female's symbol, does anyone know where that's derived from?
Isn't it supposed to be Venus?
Venus. Is Venus the god of beauty? I believe that's correct, right? And I believe that this is symbolizes a mirror. And so this is this is now, these seem like antiquated symbols, but they're the symbols that we still continue to use, but those are their origins, which is important to know. So we do this cross. Mendel does this cross, and he crosses the male pollen from the yellow-seeded plant with the female ovules from the green-seeded plant, and he produces a plant that we'll call the first filial generation, and we'll abbreviate that as the F1, okay? And this plant that he produces is a yellow plant. So we've taken the two parents, the parental strains, produced an F1 by doing this cross. This is an outcross, and we generate a yellow plant. And then he does two things, a yellow-seeded plant. The first thing he does is he takes the yellow F1 and he selfs it. So that is, he mates it to itself by taking the pollen from that, that plant and applying it to the ovules of that plant. And then he produces, so that we can indicate F1 crossed with F1, and he produces the second filial generation, which we'll just abbreviate as the F2 generation. And then he counts the proportion or the numbers of plants that he gets back that have both yellow seeds and the number that have green seeds. And when he does this, and this is where the fastidiousness of Mendel becomes really remarkable. So he didn't just go and look at a few plants. He looked at hundreds to thousands of plants. And when he counted them up, he saw that he had 6,022 that were yellow, and he had 2,001 that had green seeds, okay? And so let's just review this cross. This is very interesting. We've gone from these parental lines that breed true, generated an F1 that is yellow. So we had a yellow and a green seeded plant first. We generate a plant that has only yellow seeds. We take that plant now and we self it, and we get back both our green seeded plants and our yellow seeded plants, and we get them back with these numbers. And of course, if you do the math, what does that ratio correspond to?
It should be like 3 to 1, I think.
It'd be like 3 to 1. Good enough. Monastic work. 3 to 1. Or we can say 3/4 are yellow, 1/4 are green. He also does a similar experiment at the same time. He takes the yellow F1 and he crosses it back. This is called a back cross now with this green parental strain that breeds true. So with the green parental strain, and in this case, he's a little lazy. He gets back both yellow seeded plants and green seeded plants. And in this case, he counts about 200 of these and about 200 of these, which of course is a ratio of 1 to 1. All right, and so let's look, he did this, you know, the remarkable thing about Mendel is that he did this experiment over and over again with these 7 traits that he looked at. And so he did this trait, as I said, with plants that had yellow seeds and green seeds. He got back, he counted a total of 8,023 and noticed this ratio of 3 to 1. He did the same thing with round versus wrinkled seeds and saw the same ratio. With flower color, he saw the same ratio. With the pod shape, with the pod color, with the flower trait and the stem trait. And in all cases, he saw remarkably the same ratio of 3 to 1 in the inheritance of the trait. Okay, and this was extremely striking. Okay, but Mendel was a real scientist and a real thinker. So he has the data, but now the real advance that Mendel made is that he then thought about a model to explain the data. And let's look at Mendel's model. So for this model, he comes up with a hypothesis. And so Mendel's hypothesis is that there are 2 factors. And this is the terminology he used, auf Deutsch, Faktoren. And he said the following. He said, well, let's imagine that in the parental generation, there are 2 factors, and we'll use big Y to indicate those 2 factors. And the yellow seeded plants have 2 copies of this big Y factor. And in the green seeded one, we'll use the lowercase y. It has 2 factors as well, but they're identical in this organism. And then in the first filial generation, now we have crossed these 2 individuals, and that first filial, or the F1 plant, now has a big Y and a little y. And then the 2 experiments that he's done, in this case, for the F2 generation, he's crossed the big Y, little y plant with the big Y, little y plant. Here is the model. The model is that these factors separate when the gametes are formed. And so we can think now, we can draw this in the following way. We can consider the male gametes, and when those gametes separate, the male gamete can either have a big Y or a little y, and that happens with equal probability. So the male gametes here, half of them are big Y and half of them are little y. And similarly for the female gametes, half of them are going to be big Y and half of them are going to be little y. And then they come back together in the offspring. And so now the offspring is either big Y, big Y, big Y, little y, big Y, little y, little y, little y. And then Mendel says these factors, this combination corresponds to the following trait values. So these individuals are yellow. These individuals have yellow seeds, and it's only these individuals that have the green seeds. And so this would correspond to our 1, 2, 3 to 1, 3 to 1 ratio. And this would explain that, or 3/4 being yellow. And similarly, this model would explain his second experiment, in which he has taken the yellow F1 individuals and back crossed them to the green parentals here. In this case, we would have the yellow F1 individuals cross them with the green individuals that have 2 copies of the little y factor. And in this case, again, they would independently assort in the male gametes. So we can have half of them have a big Y and half of them have a little y, whereas in the females, half of them have a little y and half of them, the other half have a little y. And in this case, when they come back together in the zygote, we have the following combinations. And these individuals are going to now be yellow, yellow, green, green, and we have our ratio of 1:1. So central to Mendel's model is that we have what we now understand to be 3 different genotypes. Those genotypes can either be big Y, big Y, big Y, little y, or little y, little y. But we only have 2 possible phenotypes, either green or yellow. But what I want to make sure we establish right at the beginning is some common language and the language that we use as geneticists. So Mendel talked about a factorum, a factor, and so we now use the term gene for a hereditary factor. We talk about phenotype as the form taken by a character. So phenotypes in this case are the yellow seed or the purple flower, or the color of the character being the seed color, or the character being the flower color, and the phenotype is the particular value in that case. The alleles, an allele are the different forms of a gene. So when I talk about capital big Y, little y, those are different alleles of the same gene. We now, as molecular biologists, we know that these are different molecular variants of the same gene element that's encoded in the genome. We talk about dominant alleles. So this is the allele that literally dominates in the expression of the phenotype. And so we use the symbol capital Y or a capital letter to indicate the dominant allele. And a recessive allele is the allele that is dominated, and we use the lowercase. Gametes are the sperm and the egg, and they only contain 1 allele. And they are, of course, generated through the process of meiosis that you've all learned about. And the zygote is when the gametes come back together and form a fertilized egg. So this term zygote refers to the combination of the 2 gametes. And then we talk about a homozygote is one that contains a pair of identical alleles that's been inherited from the male and the female gamete. Those alleles are identical. We have a homozygote. If they differ, we have a heterozygote. And so these combinations of alleles we refer to as the genotype. So we can talk about either being homozygous recessive, homozygous dominant, or heterozygous with respect to a single gene. And so throughout the rest of the course, we'll be using this terminology. And so these are terms that you want to become familiar with so that we have a common language that we can talk about. And so this series of experiments and the observations made by Mendel and the model that he presented has subsequently come to be known as Mendel's first law, or the law of equal segregation. And so this states simply that when the gametes are formed, so that is the male or the female gametes, the 2 members of a gene pair, so the 2 alleles separate so that a gamete only receives 1 copy of the gene and only 1 allele, okay? And this is the law, and it's called the law of equal segregation because the underlying principle is that the probability that a gamete gets either a big Y or a little y from a heterozygous parental is equal. That is, it's equal to 50% in both cases. Of course, there are really interesting exceptions to this, and but that is the Mendel's first law, okay? It's interesting historical note that Mendel, never came up and never said, this is my first law. It's the law of equal segregation, but this is subsequently become known as Mendel's first law by convention. Now, Mendel, amazingly, didn't stop there. Let me just address the question. So the question in the chat is the concept of dominance recessive non-controversial, i.e. are these ideas still agreed on today?
Yeah, maybe to clarify, like what I meant is it, these seem like kind of simple relationships, like it's either dominant or it's recessive.
Yep.
Is there any middle ground?
There is that's a good question. There is, you're right, that it is a simplification of the situation where there are certainly complications, and we'll learn about some of those complications. There are really interesting cases of incomplete dominance, co-dominance. We'll learn about that in the course, and that's been subsequently, those are important concepts that we need to cover. But today, for Mendel's experiments, we're going to keep it simple. And this is, the beauty of a model is not that it explains all possibilities, but that it explains the majority of scenarios. And so that is the beauty of Mendel's experiments and his discovery, is that it explains, or it's applicable in so many different situations. So he showed that it's applicable for many different traits for which he had judiciously chosen those traits. But of course, there are traits where it doesn't hold, and we'll learn about that. But dominance and recessiveness is really key to are key genetic concepts that we need to understand. Now, what would you do if you're Mendel? Well, maybe publish your results and get a big splashy paper and go on book tours talking about this amazing finding that you found, this principle in the model that explains this, or go back to the garden and think of a second revolutionary experiment that you could do. And that is indeed what he did. And so let's talk about the second set of experiments, or experiment 2. And this is the dihybrid cross. Okay. So the first case, we called this the first experiment I called a monohybrid cross. So we have, and that refers to the fact that this F1 is typically called a hybrid. It's a hybrid of the two parentals, and there's only a single trait that we're considering, hence the mono. Now we'll consider the following scenario, which is that we'll consider two traits. Of course, these are separate traits within a plant, and so we can continue multiple. And so in this case, let's consider our parental population, or parental plants. And again, I'm just going to draw a P without it being particularly illustrative of the phenotype, but I will tell you that this P is round and it is green. And I'll tell you that this P from this plant is wrinkled and it is yellow. Okay. And now as a geneticist, we do this cross, the cross that Mendel did, and we get our F1. And when we get an F1, we get a plant that produces a round and yellow seed. Okay, so these are the phenotypes. And I'll tell you, in this previous monohybrid cross, we saw that which allele, or which was dominant, we used the symbol capital Y, and that indicated that yellow is the dominant trait, green is the recessive trait. When we do this, when Mendel did the same experiment with wrinkled and round versus as the morphology of the P, he found that it was the round trait that is dominant. And so the question for you is, given your knowledge now, the nomenclature that we have, what are the genotypes of the parent and the F1? And so why don't you take a couple of minutes to try and figure that out yourself, and then we'll come back and see if we agree. All right. I think using the same rationale, we should be able to figure this out. So perhaps it's easier to start with the F1. So let's use the symbol again. We know for yellow and green, so we know this individual is going to have capital Y, lowercase y, that's they're heterozygous for the Y gene. And this individual is also going to be heterozygous. So we'll use capital R, lowercase r. So we have two genes, each of which has two alleles. And so these parents, which again, the key point being that they breed true, the parental genotypes in this case are going to be, we know that this individual is green, is recessive, so that must be lowercase y, and this one's going to be uppercase Y. And then you can tell from the F1, you can infer what is the dominant trait, and the dominant trait, of course, is round in this case. So we will use this capital R symbol, and lowercase r for this symbol. Okay, so now we have a more complicated scenario. We have two traits and we have two factors or two alleles. We'll now use that terminology for these two traits, for these two genes. And now the experiment we're going to do is we're going to take the F1 and cross it with the F1, so do a self, in order to get the F2 generation. And now I won't draw the P's, I'll just tell you the numbers. So from this F1 self cross, Mendel gets back seeds that are round and yellow. He gets back seeds that are round and green. He gets back seeds that are wrinkled and yellow, and seeds that are wrinkled and green. So all possible combinations of these parents, these parental traits, are observed in the F2 progeny. And then again, he counts the number of these individuals, and he has, in this case, 315 of these individuals, 108, 101, and 32, for a total of 556. And now who knows what this magical ratio corresponds to?
9 to 3 to 3 to 1.
9 to 3 to 3 to 1, we say. 9 to 3 to 3 to 1 is the ratio at which these phenotypic combinations are observed in the F2. Okay, and so the question then is, what underlies this? Is this consistent with Mendel's hypothesis that there are two alleles for each gene? And so we have this ratio that we observe from the dihybrid cross. And the question is, how does this ratio arise? And so we can use just the simple laws of probability to see whether this makes sense. So one way we can think about this is thinking about random combinations of two different ratios of 3 to 1. So if we think about our offspring and we think about one of the phenotypes, we know from our monohybrid cross set, 3/4 of them, 3/4 of the F2 will be round. And of those, 3/4 will be yellow seeds and 1/4 will be green. If we think about the round versus wrinkled, 1/4 of the F2s will be wrinkled. And then again, 3/4 of them will be yellow and 1/4 will be green. And in this case, we just need to multiply along these arrows to get our probability of each of these combinations. So if we multiply 3/4 by 3/4, we get 9/16. If we multiply 3/4 by 1/4, we get 3/16. 1/4 by 3/4, we get 3/16. And again, here we get 1/16. And indeed, we have this common denominator of 16, and we have our ratio of 9 to 3 to 3 to 1. And so these observed phenotypic proportions are consistent with random combinations of two different ratios of 3 to 1. And so that's one way of thinking about it. The other way we can think about is we can think about the gametes and what is happening there. If we consider our first law, our law of independent segregation. So in that case, what's happening with the gametes when the F1 gametes are formed? Well, half of them are going to have the big R allele. So half will be big R and half will be little r. And of when these gametes are formed, if they're independent, then half will have the big Y allele and half will have the little y allele. Half of these will be big Y, half will be little y. And so again, we can just multiply along these arrows, multiply a half by a half, which will give us 1/4 are going to be big R, big Y. 1/4 of the gametes are going to be big R, little y. 1/4 will be little r, big Y, and 1/4 will be little r, little y. And those are going to be our gametic combinations in the F1. And so now if we think about the gametes in our cross, we think about the parentals. And if we have our strain that is round and green, cross to our strain that is wrinkled and yellow, we form our F1 now, which is going to have one allele from each parent and is going to be round and yellow. And now we think about the gametes from this F1 that we can form. We're going to form each of these gametic combinations up here with equal probability. And we can look at what happens then using, again, considering the male and the female gametes and all possibilities. 1, 2, 3, 4. 1, 2, 3, 4. And we have 1/4 of them will be as follows. And this is called, this way of looking at the gametes and how they come back together in the zygote. What's this called? Has anyone ever drawn a diagram like this?
A Punnett square.
A Punnett square. Many people hate the Punnett square. How do we feel about the Punnett square in this class?
It's efficient.
It's, yeah, I think it, I don't have a problem with the Punnett square, but it is a bit tedious but you can certainly see it. And the beauty of it, I think, is that it's really amenable to bingo. So for example, so I'll draw one example. So in this particular zygote that's going to be formed by this male gamete here and this female gamete, we'll bring them back together. So now we have big R, big Y, little y. So what's the phenotype of this plant going to be with respect to our two traits?
Round and yellow.
Round and yellow. Okay. So who can tell me, so let's call these male squares 1, 2, 3, and 4, female squares 1, 2, 3, and 4. So in which coordinates am I going to get a, let's see, a wrinkled yellow zygote?
Row 4, column 3.
Row 4, column 3. Let's check this. We're going to get R, R, capital Y, little y. So that's going to be wrinkled and yellow. Excellent. Who can tell me what is the most unusual, where is the most unusual zygotic combination going to occur on this box in our Punnett square?
Row 4, column 4.
Right.
There it's going to be round or wrinkled and green.
Wrinkled and green. Very special. What do we like about this particular individual zygote that's formed here? This is our double homozygote, it's called, right? Homozygous recessive. Double homozygous recessive. And you can see we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 possibilities. And this is our one single individual zygote that's going to be have the wrinkled green phenotype because it inherits both recessive alleles from both parents, okay? And so you can fill out the rest of this box at home and see that you get your 9:3 to 3:1 ratio when you consider the independent assortment of these alleles from these two different genes in the parents, okay? This is we call this the double recessive. All right, and so Mendel's, this set of experiments has been summarized as Mendel's second law, which is the principle of independent assortment. And so this law or principle states that different gene pairs assort independently in the gamete formation. And that's what we're looking at here is the independent assortment of the R gene and the Y gene into the gametes. So they have no impact on each other, and so therefore we get the random combinations of those two different genes. And so the key point here is that Mendelian genetics is governed by chance, random chance. So the probability of a particular outcome can be considered to be equivalent to the number of times an outcome is expected to occur over a large number of repeated trials. And by that we mean that each plant that Mendel looked at, that was an independent trial, and he counted them up so that he had 8,000 plants that he had looked at. Is that, yeah, 8,023 plants where he considered whether they had yellow seeds or green seeds. And then if you express that as a ratio, then that is what we call a probability. The probability, and that ratio is equivalent to the probability that a particular outcome will occur with just one trial. So if we have 3/4 is our ratio, then 0.75 is the probability that we will get a yellow P when we do an F1 self-cross and get the F2 generation. And so there's two key points or two of probabilities that you've covered before, but I just want to reiterate here. And those are the additive law of probability. So this is in this scenario in which we have mutually exclusive possibilities, A or B, yellow or green, where they are mutually exclusive, you can only be one or the other, that if we want to ask what is the realization of one or the other of the two mutually exclusive possibilities, then we add those probabilities together, the probability of A plus the probability of B. And this is in contrast to the multiplicative law of probability, which states that the probability of two independent possibilities, A and B, the probability that they are realized simultaneously is given by the product of their separate probabilities. So you multiply them together, okay? And that's what we're doing in this scenario. And so these are, principles of probabilities and how we use them to be able to talk about either two events that are mutually exclusive, that cannot happen at the same time. And if we want to ask about their combined probabilities, or two events that occur with independent probabilities, and we want to ask about them being the probability that those two independent events are realized simultaneously, and we multiply them together. I have a couple of probability questions here for you to consider. Okay, so where we have to use these two scenarios, and now we'll consider the more general case, and often we use a dice. Is dice single or die? I apologize if I'm incorrect here.
Dice is single.
Singular, yeah, and die is plural. Okay, so I'm correct. So the first question says, what is the probability of rolling a 1 or a 3 when you roll a fair dice? Okay, you can either roll a 1 or a 3. You cannot roll both at the same time. So that means that they're independent, they're mutually exclusive, right? You either get a 1 or you get a 3. And by contrast, what is the probability of rolling a 1 when you roll it the first time and then rolling a 2 on the second turn, okay? And so these are the two scenarios that you want to be able to consider. If a dice has 6 sides and we have 1 through 6, we roll it, the chance of getting a 1 is 1/6. The chance of getting a 3 is 1/6. And if we're asking what is the probability of rolling either a 1 or a 3, we add them together. And so we have 2/6 or 1/3 is what it reduces to. If we then consider, again, we roll the dice once, we get a 1. There's a 1 in 6 chance of doing that. If we roll the dice again and we ask what's the probability of getting 2, there's a 1 in 6 chance of getting a 2. And so the probability of getting a 1, then a 2 is you multiply those probabilities together, and that's 1/36, okay? And so whenever you're considering probability questions, you need to be thinking about, are these mutually exclusive possibilities? Am I looking at independent probabilities and whether we want to multiply them together? Now, you need to go and read The Monk in the Garden to learn about Mendel's life. I'm not doing you justice when I'm talking about his experiments and his life here. It was really fascinating and tragic in some ways. His study was published in 1866 to absolutely deafening silence. So, he wrote this all up. He realized that this was super important. He wrote it up in a monograph. He mailed it to all the luminaries in the field, including Charles Darwin. And it is said that if you go to Darwin's library, there is a copy of Mendel's monograph there. And in those days, when the books were printed, the pages, you had to get a knife and cut the pages open to be able to separate them. And apparently Darwin's copy, those pages are not separated, and suggesting that he never even looked at it or read it. And this generally seemed to be the case, is that Mendel's great discovery was unappreciated for many, many years, until 1900, when 3 scientists who were botanists interested in heredity at the time simultaneously rediscovered Mendel's research and instantly, or very quickly, realized the important implications of Mendel's model of the particulate basis or the discrete basis of inheritance. And so this was De Vries, Correns, and von Tschermak, who were considered the rediscoverers of Mendel, some 30 plus years after his initial findings. But that really set the ball rolling. And it was actually just a couple years later that Sir Archibold Garrod, who was a medical doctor, who was working on and studying the inheritance of diseases in families, understood the implications of Mendelian inheritance for human disease. And the particular disease that he was looking at is called alkaptonuria. This is a disease in which the urine of people who are, or the way it's diagnosed is that the urine of people who have this disease turns black upon exposure to the air. And this disease has subsequently been identified to be the result of a mutation in an enzyme, HA oxidase, which converts homogentic acid to downstream metabolic product. And so when that gene is defective and encodes a non-functional protein, you get a buildup of homogentic acid, and people have this disease, alkaptonuria. And this class of disease, which are known as inborn errors of metabolism because they affect metabolic pathways, were a class of human genetic disease in which many examples of Mendelian inheritance were identified rapidly following Garrod's understanding of the implications of this study that had been performed in peas for understanding human inheritance. All right, so today we've had a whirlwind tour through the research output of Gregor Mendel. The 2 key observations he made is the law of equal segregation. So that is that for a diploid organism, the 2 copies of the gene are going to go into the gametes with equal probability. And then the principle of independent assortment, which is that 2 genes are going to assort the different alleles into the gametes independent of each other. And we have revisited the laws of probability, the additive law of probability and the multiplicative law of probability, in order to understand how these laws result in the observed phenotypic proportions.